Barley is an important crop in the U.S. and is cultivated on over 3 million acres annually. Its primary use is for animal feed, but a substantial portion is used in the malting and brewing industries. Today, the U.S. barley crop is threatened by a serious disease called stem rust, which is capable of destroying fields within a few weeks after they are infected. Stem rust is caused by a fungus and is named for the reddish-orange pustules that erupt from the stem. On the northern Great Plains of the U.S., stem rust has been kept in check for over 50 years by breeding varieties that resist the disease. However, all U.S. varieties are vulnerable to infection by a new and highly virulent race of the stem rust pathogen from East Africa. Scientists call this new stem rust race Ug99 because it was first discovered and characterized in Uganda in 1999. But stem rust spores can be easily disseminated by wind over hundreds of miles and still survive to cause disease. Because of this, race Ug99 and its variants have spread across eastern and southern Africa, traversed the Red Sea into Yemen, and are now established in the Middle East. Rust spores also can be moved by people traveling across regions. Thus, it may only be a matter of time before race Ug99 reaches the U.S. The Barley Coordinated Agricultural Project, or Barley CAP for short, is a community effort of scientists from institutions in 13 different states focused on identifying genetic markers or tags that will dramatically speed breeding efforts to improve barley. Barley CAP has partnered with the USDA Ug99 Cereal Rust Initiative to launch a major offensive against this new disease threat. Finding genetic markers associated with genes for Ug99 resistance is a priority for Barley CAP researchers. To identify resistance in barley, researchers exposed thousands of U.S. breeding lines to Ug99 in Kenya, where the race is present and conditions for stem rust infection are favorable. Although most lines were susceptible to the disease, a few showed high levels of resistance. Discovering resistance already present in advanced U.S. breeding lines makes producing a commercial variety with Ug99 resistance easier, since there are fewer genetic differences than between, for example, an unadapted line. Barley CAP researchers have completed a roadmap of the genetic markers of barley. This roadmap can be used to locate genes controlling economically important traits, like stem rust resistance. Indeed, 2 genetic markers were found to be associated with Ug99 resistance on a specific barley chromosome called 5H. These genetic markers are now being used by breeders to select lines that carry the resistance genes. The ability to indirectly select for genes controlling Ug99 resistance provides a real advantage. Using these genetic markers makes it more rapid and convenient to develop resistant breeding lines. Also, it does not require having to infect plants with the pathogen, which in the case of Ug99 is critical since it cannot be brought into the U.S. for field testing because of its potential destructiveness. In summary, Barley CAP researchers have developed the resources needed for breeding superior barley varieties more efficiently. These tools can be effectively used to address new disease threats to the crop. For example, the genetic markers that were just described as associated with Ug99 resistance are being used to speed the breeding process, so barley varieties with stem rust resistance can be released to growers before widespread crop losses occur.